KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
Free bus for LKG to 8th standard.
Free, hygienic and nutritious food.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Now, the processes of recombinant DNA technology. In process of recombinant DNA technology, first side heading, that is isolation of genetic material or the isolation of DNA. So this is the first side heading. So there, what is the point now? Nucleic acid. The nucleic acid is a genetic material of all living organisms. So that's we know very well. So definitely nucleic acid is acting as a genetic material in all living organisms without any exceptions. So that point directly only we can understand. So but here what is the point? In a majority of the organisms, in a majority of the organisms, DNA is a genetic material. Why? Because here our knowledge concern nucleic acids are two types are there. One is DNA and one more is RNA. So means definitely in all living organisms either DNA or RNA must be there. Means here in all organisms or all living organisms definitely we can find the presence of nucleic acid, but most of the living organisms, in most of the living organisms, deoxyribonucleic acid or the DNA is genetic material. So in most of the living organisms, genetic material is DNA or deoxyribonucleic acid. Nucleic acid. Now, in order to cut the DNA, so in order to cut the DNA with the restriction enzymes, in order to cut the DNA with restriction enzyme, it needs to be pure form. Means here definitely, so if you would like to cut the which one DNA with the restriction enzymes, no, so definitely only DNA only you have to take. So we should not take RNA or other kind of macro elements, we should not take. Definitely pure form of DNA only is required. Pure form is nothing but, so there RNA or other kind of proteins or any kind of macromolecules should not be there. So in order to cut the DNA with the restriction enzymes, it needs to be the pure form. Okay, pure form free from the other macromolecules, I said or not. So means here definitely pure form of DNA definitely is required, and there is no chance they should not or we should not maintain any kind of macromolecules along with the DNA at the time. Now suppose if along with the DNA, suppose any kind of macromolecules are there, means at the time we should not call that particular that particular thing as pure form. Means there only DNA must be there and that must be free from other macromolecules. So since the DNA is enclosed with the many membranes, so there are different types of membranes are there. Suppose if you take outermost membranes, suppose cell wall will be there, within the cell wall cell membrane is there, and here within the cytoplasm again nuclear membrane is there. Likewise there are different types of membranes are there. We have to break the cell to we have to break the cell open to release the DNA. So means in this particular case, definitely in order to obtaining of or in order to get the DNA, definitely we have to break the different types of membranes. So that is information they are giving. So to open the release of or release DNA along with the other macromolecules means, first of all, so in order to obtaining of DNA, so when we are going to cut the membranes, so there are different types of membrane also are there, along with all membrane or along with all macromolecules, DNA also will be there. Now what we have to do is, so these macromolecules, all these macromolecules except DNA, we have to digest. So there, first of all, let us see what are the macromolecules are present in the cells means, roughly. Now let us take RNA will be there, proteins will be there, polysaccharides we can see, and lipids also we can see. Means RNA, proteins, polysaccharides, lipids. Likewise, there are different types of macromolecules we can be seen. But all these macromolecules, now we no need to take which one we needed. Pure form of DNA is needed. So in this particular case, now this can be achieved. So how we can be obtained means the treat of so now here, if you would like to do the experiments, if you would like to do the experiments in bacterial cell, so or we can do the experiments in plant cells, or we can do the experiments in animal tissues, so wherever you want to do, so there we can be done. So how, suppose if you would like to do the, or if you would like to take the bacterial DNA means, so here bacterial cell wall must be digested. So here you see there, bacterial cells digested by the lysozyme enzyme, and one cellulase, ASCM, cellulase definitely we will be used for the breakdown of plant cell wall, and chitinase definitely we will be used for the breaking of fungal cell wall. So likewise, there are different types of enzymes are there. One more time, lysozyme enzyme for breaking of bacterial cell wall, cellulase enzyme for the breaking of plant cell wall, and chitinase enzyme for the breaking of fungal cell wall. Likewise, there are various types of enzymes are there which are involved in the breaking of cell walls or different types of cell walls. So now here, so when we will be cutting, initially just we are cutting the all cell walls. So there definitely when we will be removing the cells, at the time, so what are the contents, or what is the content is present within the cell, definitely that will be coming, that will come out. So in that particular case, that particular thing definitely consists of macromolecules. Along with the macromolecules, DNA will be there, so that is information. Now you know that the genes are located on the long molecules of a DNA intertwined with proteins such as histone proteins. So this one really only we know. So why? Because this explanation already I have given in a molecular basis of inheritance. So means what is the information they are giving? So here DNA or the genes are located along with the molecules of DNA intertwined with the proteins known as histones. So their histone proteins are there. So long molecules of, why? Because here in packaging of DNA helix we have seen histone proteins. There are different types of histone proteins, H2A, H2B, H3, H4 likewise. So we have seen there. So definitely for intertwining of which one? So definitely for the circular or for the coiling of DNA, definitely which one is required? Histone proteins are definitely required. So that point we have seen. Now RNA can be removed. Now here there are different types of macromolecules are there now. So there are different types of macromolecules. Now let us concentrate on this one. So RNA can be removed, or RNA is can be removed by the treatment with the ribonuclease. So here in order to removal of, or in order to removing of RNA, so what is the enzyme we have to use means ribonuclease enzyme. Next one, whereas proteins, suppose if you would like to digest the proteins, so here we have to use the protease enzyme. Next one, other molecules means here lipids also are there, no? So here they didn't give information regarding lipids, but lipids can be digested with the lipase. Lipase enzyme. So likewise, next one, other molecules can be removed by appropriate treatments. So here means suitable treatments they are asking to follow, but what are the techniques names? They didn't give. But suppose in order to, or if you would like to remove other kind of molecules from that entire sap or liquid, so if you follow appropriate techniques, definitely, so we can remove all kinds of materials. Now at the time, definitely we can obtain pure form of DNA. My dear students, here one point you have to remember. That point is, so here DNase enzyme you should not use, or deoxyribonuclease enzyme we should not use. Suppose if you use DNase or deoxyribonuclease, means what will be happen there? DNA also will be digested. If DNA is also digested, means then what you will isolate? So did you understand? So that's why here we will use all types of enzymes for removal of all types of macro elements except DNase and except deoxyribonuclease, ASE. So deoxyribonuclease. So why? Because so when you will digest all these macro elements or the macromolecules by using of different types of enzymes, then only we will find purified form of DNA. So this is the thing is required. Sir, if you obtain this purified DNA, then what you will do, sir, means now ultimately the precipitates out, means finally, so we will observe the formation of precipitation. How it will be known? So exactly when you will suffer from the cold, when you will suffer from the cold, no, so you will sneeze, you will sneeze at the time, no, you will see the material. So in your hands exactly same color, same like that only. So purified form of DNA we can be observed, or so you can be obtained. So that kind of thing only, so we will be studying after the addition of chilled ethanol. So this kind of a purified form of DNA ultimately precipitates out when definitely we can be see the formation of purified form of DNA in the form of precipitation. So after the addition of chilled ethanol. So chilled ethanol, so here they didn't give clearly. So but our practical is concerned about how we will add this chilled ethanol on the walls or at the walls of the test tube. So on the walls of the test tube, when we will like a drop by drop we will release the, or we will pour the which one chilled ethanol at the walls of the test tube. So in that particular case, definitely purified form of DNA ultimately precipitates out. So we have seen, so we have done, we done this experiment, means not in the presence of you, for your seniors I have given explanation. So when you people will come to the lab, that time I will explain. So this one isolation of DNA in our laboratory conditions. Now here up to here, okay, now the last point also is there, that can be seen as collection of fine threads. So here there we can be seen like fine thread like structures we can be seen in suspension. So likewise some more important points they have given. Now let us have a look on this information. So that is isolation of DNA or isolation of genetic material. First of all, so here what they have given, no, so there are different types of macromolecules are there. So all these macromolecules we have to digest. So if you would like to get the purified form of DNA, so in this particular case, we can by using of our technological things, so we can remove all different types of macromolecules. So suppose if you would like to remove the RNA ribonuclease enzyme, and if you would like to remove the which one proteins, protease enzyme, and if you would like to remove the lipids, lipase enzyme, and likewise by using of different types of appropriate methods, definitely we can remove the different types of materials which are present in the cytoplasm as well as nucleoplasm. So then, so definitely we can find out, definitely we can get the purified form of DNA ultimately. So that is precipitates out. So when once if you add chilled ethanol at the walls of the test tube, so we have to use only chilled ethanol for the obtaining of a pure form of DNA. Next one, so how it will be means, so this particular DNA will be like fine threads. So like thread-like structures we can be seen in entire precipitation. So this particular thread-like, this fine thread-like structures, so we can be separated by using of spooling method. So spooling method definitely we will be using for the separation of DNA. So DNA, so means in order to obtaining of DNA, what is the method generally we will follow, means spooling method we will follow. So likewise definitely we can obtain pure form of DNA as fine threads. So that particular thing we can be separated by the spooling method. Next thing, what is the cutting of DNA at specific locations. Now we have the DNA. We have a DNA. Now we obtained no. So by using of a first step, we have obtained. We obtained that particular DNA. So that particular DNA, what we have to do is we have to cut. So at specific locations. So to cut at specific locations, definitely now we have to take the help of. Can you guess? We have to take the help of. In the first line only I have written which one is required. Restriction enzyme is required. Definitely restriction enzyme or restriction endonuclease is there now. So restriction enzymes two types. One is exonuclease and one more is endonuclease. Now there restriction endonuclease enzyme definitely we will use. Now restriction enzyme digestions digestions are performed by the incubating purified DNA molecule with the restriction enzyme. Means here what we have to do, restriction enzyme as well as pure form of DNA we have to keep for few days. Means combinedly like those mummy how we will prepare like rice and rice flour as well as one more. What is that like black gram. Yes, so how mummy will put no. So likewise these two means one is restriction enzyme as well as purified form of DNA we have to keep for incubation. So in that particular case what will be happen no. So at optimum conditions, when by keeping off these two at one place, so when you will maintain optimum conditions for this specific enzyme, so definitely that particular enzyme will cut this DNA into fragments. So now we will be getting fragments. So at the time, sir, we have first of all in first step we obtained purified form of DNA. Now by using of restriction enzyme, now we have cut the DNA into fragments. Now what we have to do, sir, means now so gel electrophoresis method means agarose gel electrophoresis method we have to employ. So means why, sir, this one means already I have given an explanation. So this gel electrophoresis definitely helps for the separation of DNA fragments based on the molecular weight. So when we will be or we can separate the DNA fragments based on the molecular weight, so by using of a technique known as gel electrophoresis. So in this particular case now I asked to remember gel electrophoresis. Technique in four different which one? So four different which one? Four different side headings I asked to remember. So what are those? Now you see there. So first of all, ethidium bromide is required. Ethidium bromide. Next one like UV light, and one more is bright orange color, and one more is yellowish. Likewise, there are different types of side headings we have to remember under the gel electrophoresis. So here in a gel electrophoresis technique. In a gel electrophoresis technique, definitely we will use agar-agar gel. That is only we have obtained from the red algae. So next one, so this gel electrophoresis is employed to check the progression of restriction enzymes digestion. Next one, the DNA is a negatively charged molecule. So this point we have learned in so many times. So we have seen in so many times it is coming. So thereby, so here a DNA is a negatively charged molecule. So because of the presence of which one? Phosphate. So here because of the presence of a phosphate, DNA getting negative charge. So don't forget, hence it moves towards the positive charged electrode anode. But here, so here what is the information now? Already this information everything we have learned. So this process is repeated with vector DNA. So means here not only this entire means the process of gel electrophoresis we have used for the separation of DNA fragments based on the molecular weight or based on the length of DNA we can separate. Means same kind of process, same kind of gel electrophoresis process we have to use for the vector DNA also. repeat. This process is repeated with the vector DNA also. Don't forget this point also is important. Means the electrophoresis process we will done not only for the DNA separation of DNA fragments, definitely we will use for the vector DNA also, with a specific restriction enzyme. The cutout gene of interest or desirable gene or foreign gene or alien gene, likewise, or 4 or 5 names are there. Gene of interest from the source of DNA and cut vector with the with space or mixed. Okay, mixed and ligase is added. So here what is information now? So by using of restriction enzymes, we have to cut the both gene of interest as well as plasmid or vector. So plasmid or vector, and one more is gene of interest or where we will find desirable characters. Now that particular location we can be taken as gene of interest. Means here one point I ask to remember, that point is, so what is the name of the restriction enzyme we are using for the cutting off for the cutting of DNA? Now say same kind of or same type of restriction enzyme only you must use for cutting off plasmid also. Don't forget, same kind of restriction enzyme we have to use for the cutting off both sources. So when this particular desirable gene or gene of interest we will be placing on or we will be placing in plasmid, at the time definitely we can be seen the formation of recombinant DNA. Already I told you over 4 or 5 times, recombinant DNA is nothing but the combination of 2 different types of genes together. So there one is a plasmid DNA is present and one more is newly inserted gene of interest or a desirable gene or foreign gene or alien gene. Likewise there are different types of names are there, but content is the same. So means here recombinant DNA, how this recombinant DNA formation takes place means by the addition of a foreign gene with the plasmid. So definitely plasmid or vector, definitely recombinant DNA formation takes place. Now suppose, one explanation is there, that is also important. That explanation is amplification of gene of interest, amplification of gene of interest using PCR. So this one, so if you are going to write public examination, so definitely this PCR technique definitely important. So try to learn very easily. I will explain for you. So try to listen carefully. Now amplification means Xerox. So Xerox, amplification means multiplication. So what is the mean multiplication or the amplification of a gene of interest? Means gene of interest means desirable gene is there, no? Gene of interest or a foreign gene or the gene which has good characters or novel characters. So that one we can be multiplied so by using of PCR. PCR means here I have written PCR stands for polymerase chain reaction. So polymerase chain reaction. So in this particular case now, in this reaction multiple you see there, in this reaction multiple copies, multiple copies of gene or the DNA of gene of interest is synthesized in vitro conditions. So here once you see that, so in amplification technique by using of PCR, what we will do, sir, generally means definitely we will do that multiplication of gene of interest or the multiplication of a desirable gene. So we will be synthesized in laboratory conditions. Sir, laboratory in vitro. In vitro is nothing but laboratory conditions. So using of 2 sets of primers. So now don't forget. So here how many number of primers we will use means 2 sets. 2 sets of primers we will use. Now the small, so primers are nothing but, what is the meaning of a primer means small chemically synthesized oligonucleotides that are complementary to the regions of DNA. So one more time, the small chemically synthesized oligonucleotide that are complementary to the regions of DNA. Okay, now you try to take that one. So primers are nothing but, or simply you can remember small chemically synthesized oligonucleotides. So that one we can be taken as primer. Now and And what are the things are required along with the primers? Along with primer, enzyme. One enzyme is required, that is DNA polymerase enzyme. In a molecular basis of inheritance chapter, no. So we learn no, DNA polymerase definitely helps in a polymerization from 5' to 3' direction. Likewise we see no. So that one, so same kind of enzyme is required. So that particular enzyme name only DNA polymerase. The enzyme extend the primers using nucleotides. So definitely, so here the primers now only we learn no, with a small very small oligonucleotides, so but all these are all will be extended. So extended, where it is extended, here extends, so by using of DNA polymerase enzyme. Okay, now and genomic DNA as template. So here, if the process of replication of DNA is repeated for many times, the segment of the DNA can be amplified to approximately billion times is achieved by the use of thermostable DNA polymerase, means isolated from the bacterium, that is Thermus aquaticus. Now here let me explain that information. What is that now? So now, so here we will be using, so in a PCR technique, what is the name of the enzyme we are using? No, thermostable means thermostable DNA polymerase enzyme we are using. This is thermostable DNA polymerase enzyme. What is information of this one? No, definitely this particular enzyme we have been isolated from the Thermus aquaticus. So this particular bacteria where it is present, no, this particular bacteria is present in like hot springs, like in a Discovery Channel we will see, no. So in that particular area, no. So hot springs means the area which will have hot water. Likewise, that will be coming out from the soil. So such kind of atmospheric conditions, our scientists so isolated Thermus aquaticus bacteria. From that Thermus aquaticus bacteria, they have isolated this one, tag polymerase or thermostable DNA polymerase. Tag polymerase or thermostable DNA polymerase, both names are same. Next one, so amplified desirable gene is ligated. So what is a desirable gene is ligated? No, ligated with a vector. No, means here after completion of a PCR and everything, no. So what is a gene we will be obtaining? Okay, suppose if you multiply the desirable gene, so that particular desirable gene now we will join, we will ligate with the vector. So that but after recombinant DNA formation takes place, so that recombinant DNA we will transfer into the host. So likewise, so PCR technique we can explain. So now here I would like to explain PCR technique based on the diagram. So that's why here a diagram also I have drawn. Once you see there, so there are 3 steps are there in PCR, 3 steps. First one is denaturation, annealing, extinction. One more time, denaturation, annealing, extinction. In a denaturation, so approximately what is the temperature we will use means 95 degrees centigrade. Next one in annealing 55 degrees centigrade. Next one in extinction 72 degrees centigrade. One more time, in a denaturation 95 degrees centigrade, in annealing 55 degrees centigrade, and in extinction 72 degrees centigrade. Now first step is denaturation. So in a denaturation, the word itself giving information regarding denaturation. That is, so now you see there, here double stranded DNA is there. Now in a denaturation process, what will be happen now at 95 degrees centigrade? So this double stranded DNA, that is converted into 2 single stranded DNAs. So now you see there, so one is here and one more is here. So now once the structure you observe there, so first of all, when these 2 strands are present together, hydrogen bonds are in a normal situation. But so in after denaturation process, by using of 95 degrees centigrade, now hydrogen bonds are break down. You see there, here the gap is there. So that process we will be calling as denaturation. Next one, in annealing. In annealing, what is the process we will do means, don't forget, so definitely they will ask in NEET examination, that is addition of primers. Once you see there, here arrow mark is there, no, addition of primers. So addition of primers, where we have to add sir means, so exactly opposite to the 3' of old strand. So why we will add opposite to the 3' old strand of or 3' of old strand? Suppose if you add at 3' of old strand now, opposite one definitely starts with 5' now. So our knowledge concerned, definitely polymerization starts from the 5' to 3' direction. So thereby, so definitely polymerization takes place from the 5' to 3' direction. That's why definitely primers we will add at opposite to the 3' of old strand. If you add opposite to the 3' of old strand, no, definitely polymerization starts with 5' to 3' direction. That's why in a diagram, in information also we have seen addition of two sets of primers we seen. So one here means our knowledge concern, here two strands are there now, two single strands are there. So here in a third frame, opposite one primer we are adding. So this side also opposite 3' another primer we are adding. Okay, so now you see there, this strand will move like that, and here this polymerization will move like this. So in extinction, what is the point? No, extinction, the word itself giving information. Extinction is nothing but, or extinction we will do at 72 degrees centigrade. So in extinction, what is the information? No, with the help of DNA polymerase enzyme. With the help of DNA polymerase enzyme, polymerization takes place. So thereby, when the polymerization process takes place, so these two single stranded DNAs, so here in one strand, here one more strand is there, these two single stranded DNAs definitely converted into two double stranded DNAs. So here one and here one. So now you see that, now you are observing two double stranded DNAs now. So likewise, so definitely we can see the formation of two double stranded DNAs. Means earlier we have taken one double stranded DNA only, no. So but after completion of 3 steps of a PCR technique, now we are finding two double stranded DNA.